Up to this point in our discussion about nucleosides, nucleotides, and DNA, we've looked at DNA as a single-stranded molecule shown on this slide right here. But when DNA was discovered, we realized that, hey, it's not a single-stranded molecule, but it is a double-stranded molecule right here. So this is a DNA molecule, and it is composed of two strands. You can see right here, this little piece right here. In that red rectangle, what's contained in that rectangle? Well, we have the phosphates and we have the sugars. So we have a ribose phosphate backbone. And there's the first backbone, and then what's protruding off of that backbone of DNA are the nitrogenous bases, shown with the one-letter code there. And then on this side, we have another backbone right there. And so these two linear pieces, this linear piece and this linear piece, they are going to associate with one another by hydrogen bonds to form this double-stranded DNA molecule. So if we come back to this slide right here, now this is a zoomed version. I want you to see, okay, what does the phospho backbone look like? This is what it looks like, right? There's all the phosphodiester bonds right there. We have the ribose sugars, okay? And then when we go to this slide right here, we kind of don't show as much detail with the backbone because in this slide we're focusing mainly on the nitrogenous bases. And then in the next couple of pictures, you're going to see the backbone just represented as a ribbon. So I want you guys to understand like those three versions, the ribbon, a little bit of detail, and on the previous slide, a lot of more detail. So let's talk about this double-stranded DNA molecule, because when we first discovered DNA, there was a logical question. So we know that there's two backbones, and we know how the backbone is put together chemically. It goes in the 5' direction to the 3' direction. So could you have both of those backbones like this? That line right there, I'm representing that as the backbone. And apparently, we lied. So if we had a backbone like this and a backbone like that, could it be oriented in this manner? The strand right here is 5' to 3', and then the second strand 3' or 5' to 3' in that manner. Or could we have it like this? 5', 3', 3' to 5'. So this would be a parallel strand. These strands would be parallel. And then this one over here would be anti-parallel. So that was a question like, well, what is it? Well, a lot of evidence suggested that it's this guy right here. Okay? So what's some evidence that suggests that DNA is anti-parallel? Well, when we look at the mechanism of DNA synthesis and how DNA polymerase works, that's strong evidence that suggests, hey, DNA is the anti-parallel molecule here. And then, what other evidence do we have? We have X-ray crystallography. We basically have a picture of DNA, and it shows us that it's anti-parallel. Okay? You and I may take that for granted in this day and age because we've been taught this since ever, and so it's always been known. But just think about it. Just 100 years ago, 80 years ago, it was like, What is it? Right? Another thing that they noticed about a DNA molecule is that when they looked at the nucleotides here, they would always see, it didn't matter what fragment of DNA they looked at, they always noticed that the ratio of G to C, that ratio was always 1 to 1. When they compared G to A, that ratio could be all over the place. When they looked at A and T, they noticed that ratio between those two nucleotides, always 1 to 1. But when they compared T to C, they're like, it wasn't 1 to 1. So this observation made us think a little bit, like what's going on here. And you guys know that this is called base pairing. And that double-stranded helix there, or DNA, double-stranded DNA, in this graphic right here, you see how the hydrogen bonding network works. Let's clean that up a little. You notice that C pairs with G, G pairs with C, right? And then A to T, T to A. That's base pairing. And why is it base paired that way? Because of the hydrogen bonding and how it's all oriented. So when we take a look at adenosine right here, or adenine, and we look at its structure right here, okay, on this side of the molecule right there, what do we notice chemically here? We see that this oxygen atom right there has 2 lone pairs. And then we have this hydrogen right here coming off of a nitrogen. Remember, which atoms can participate in hydrogen bonding? Just remember fun. Or fawn. I just say fun. It's fun because the hydrogens have to be attached to either a fluorine atom, an oxygen atom, or a nitrogen atom. So we see that this nitrogen atom right there has a hydrogen attached. So that's the hydrogen donor, and this guy would be the hydrogen, or the hydrogen bond acceptor. And then we have this nitrogen right here in the purine ring, and that nitrogen is going to have a lone pair. You can draw on some lone pairs right there. And then on the thymine, you see that we have a nitrogen atom that has a hydrogen on it. There's the hydrogen donor. There's the hydrogen bond acceptor. And we have a hydrogen bond. Why can this hydrogen right here, why can we not have a hydrogen bond like that? Well, because that hydrogen is attached to a carbon atom. It's not one of the 3. So that is not a hydrogen bond. No, no, no. So all we have when we look at the adenine and the thymine base pair is it only has 2 hydrogen bonds. Right? In contrast, when you look at guanine and cytosine, you can see what? 3 hydrogen bonds. And I don't think it's critical that we go through that analysis again for each one, but the basics here. We have some lone pairs here. That hydrogen bond, lone pair there, lone pairs there. Okay, so what are we observing here? We're just observing how the hydrogen bonds work in the DNA molecule. Okay? So what would you expect? Which type of DNA molecule would have a higher melting point? A DNA molecule with more GC content or more AT content? Well, in the AT content molecule, we have a lot of ATs, but they only have 2 hydrogen bonds. It is the hydrogen bonds right here that it's not solely just hydrogen bonds, but what's holding those 2 backbones together, or those 2 DNA strands together? Because they could technically, right, just float away, right? But DNA is a double-stranded molecule, and it's because of these hydrogen bonds that holds the 2 strands together. And so when I talk about melting DNA, we can heat it up and take those 2 strands and separate them. And that's what we what's happening when I say, what is the melting temperature of DNA? Separating those 2 strands. So if we come back to the thought experiment that I'm giving you, you have a DNA molecule with high GC content and a second molecule with high AT content. Which one's going to have the higher melting temperature? And the one that's going to have the higher melting temperature is the DNA molecule that has the most GC content. And that is because the GC base pairs here have 3 hydrogen bonds. And so that idea of the GC content makes the melting point higher is something that we have to consider when we design DNA primers. And that conversation is going to be had when we look at PCR. Okay? Because we get in biochemistry and biotechnology, we get to design DNA primers, and we have to take into account how much GC pairs we have versus ATs in order to design a really good primer. But that's a conversation for another video. That's just giving you a little heads up of what's coming and why I'm spending so much time talking about these hydrogen bonding, because it's important for experimental design in the future. Let's consider or continue our conversation about DNA. So things that you probably already know about DNA, that we have a major groove and a minor groove. And those come into play when you have proteins that need to bind DNA. Certain proteins are going to bind in those different types of grooves. Now, some other key features here. Some numbers that are good to just have in your head. How many base pairs does it take to go from one point of the helix and make it go all the way around and complete one turn? So what you're looking at, okay, let's pay attention to just one of the strands. Okay, and I'm going to trace this one right there. Okay, so that strand that I traced in red, we see that it's in the very front, right here. Okay, and when we go and make a turn, where's that strand? It is behind. It's behind this front strand, and it keeps coming, and now there's the front again. Because this is a helix, DNA isn't just like we showed before, where it's just lines like this, 5' to 3'. Okay? It's not like that, but it's a double-stranded helix. And this double-stranded helix has orientation to it. It is going to be a right-handed helix. So we have a right-handed double helix here. So what else? So that how many nucleotides does it take to go a full turn in our DNA helix? Well, it's going to take 10.5 base pairs is what we call it. So 10.5 base pairs to go a full turn in a DNA molecule. And that full turn you can see is 36 angstroms. Okay. And then we have right here this number, the distance between two base pairs is 3.6 angstroms. And then you can also see how wide is that DNA helix. That's about 20 angstroms wide. So those are some key numbers to understand and remember. Now, I just traced the backbone, okay, shown by that blue ribbon. Now we're getting more cartoonish. I'm not showing you the chemical structures here. But looking at this version of DNA versus this version of DNA and this one right here, all showing a little bit different things. With the DNA backbone, let's look at this molecule here. We see all the blue atoms and a touch of red in there. Okay. That blue part on the outside of the DNA molecule, so like right here and right there on the outside of the DNA molecule or on the backbone, what is the charge? Is that going to be neutral, positively charged, negatively charged? Well, if you go back and look at the chemical structures of the backbone, you know that you have phosphates, and those are going to have negative charges. So the backbone on the outside of the molecule right here, all on the outside, it's negatively charged. Right? When we look at these yellow bars, what's that representing? That's representing the nitrogenous bases, and you can see the hydrogen bonding here. Now, these hydrogen bonds in this graphic right here is not showing you if there's 2 hydrogen bonds or 3 hydrogen bonds, because we don't know the base pairs here. It's just a generic, there's a nitrogenous base. Just double checking here. And there's something that we're going to talk more in depth in class about the stability of a DNA molecule. We've already talked about the hydrogen bonding network between the bases, and that's going to hold the DNA molecule together. But what else we have is called base stacking. When you take a look at this structure in the middle and look at the nitrogenous bases right here, they are pretty much planar. Not perfectly planar, but the way it's drawn, you can see how they're kind of flat. They're mostly flat. So the base stacking happens that, hey, the base above and below, they can stack on one another, not like right on one another, but there's going to be a interaction between this nitrogenous base and this nitrogenous base, not by hydrogen bonding, but by pi stacking or base stacking. Okay? And so we're going to talk a little bit more about that in class, and And so that's another way DNA is stabilized. Okay. And you can kind of see that with this graphic here. But we'll go into more detail about that in class. The next thing that I want to talk about in regards to DNA is that DNA can have different forms. As you can see in the slide, we have the A form, the B form, and the Z form. Okay? Now, both the A form and the B form are right-handed helices. But the Z form here is a left-handed helix. Okay? So let's first focus on bDNA. bDNA is what you're going to find in human cells. That's what we are used to seeing. That's what we talk about all the time. Okay? And so everything that we've discussed about like how many base pairs does it take to complete a turn, 10 and a half, all those numbers refer to the B form. Now the A form is basically coming from the B form. It's just in the A form, it looks like that when you get rid of water. When you put this DNA in other solvents other than water and get a crystal structure of it, you see that it's going to look a little bit different. And I like looking at these graphics here at the bottom here. You see the B form, all the nitrogenous bases and how they're oriented in the middle there. But then in the A form, it's more closer to the phosphate backbone. And so that's what we see DNA looks like when we get rid of water. Kind of interesting. Now the Z form is drastically different than A and B in the fact that A and B are a right-handed helix. The Z form is a left-handed helix. And scientists have found the Z form in bacteria and a few eukaryotes. And honestly, we don't really know why. Why do some species have small fragments of DNA in the Z form? Don't know. Still trying to figure that out. But those are the two forms, or the three forms of DNA that I want you to be aware of that exist. And this table here kind of like puts it all together. So if I ask the question, between A and B form, which one has a bigger diameter? Which one's wider? Well, you see right here that the A form is a whole lot wider than the B strand, or the B form. Okay? You can see all that, you can compare and contrast all the numbers there. Okay? So the Z form, you can see that it's getting more slender, okay, because that diameter is getting smaller and whatnot. Another thing that you see here is the carbon 3' endo. That, just like how we talked about in proteins, where you have the proline, and that proline has the gamma carbon, and it can be puckered endo or exo. And depending on what protein that you're talking about, so for example, collagen, the cells take proline and they tack on a hydroxyl group at the gamma position, and that hydroxyl at the gamma position makes the gamma carbon puckered in the exo form. And we need that proline derivative to be in the exo position in order for collagen to have its physical properties. And so the same thing with DNA. You have a 5-membered ribose sugar, and you can have that ring that puckers. And we're going to talk in class more in detail what that is and why that's important. But in this table, you can see another difference between the A form and the B form. In the A form, you want the 3' carbon to be endo. In contrast, in the B form, did I say that? In the A form, you want the 3' carbon to be endo, and in the B form, you want the 2' carbon to be endo. Okay? You notice that, what? None of those are in exo. Isn't that kind of weird? Like collagen, we wanted the proline exo, but in DNA, we want the ribose sugars to be endo. Right? What's another interesting thing here? What do the A and B forms have in common, though? That the glycosidic bond has to be anti. Remember, that's where the base can be rotated, anti or syn. But zDNA, look at that. It prefers some, for the purines or pyrimidines. No, wait, what is that saying? Syn for purines. Purines are what? The nitrogenous bases with the 2 ring systems, right? And so those like it syn. It's just kind of weird. Let's see. When you look at the Z form there, look how just crazy it looks, right? But all those little differences, right here in this table, you can see all those little differences all culminated together has a drastic effect on the shape of the DNA molecule. Kind of cool to me. Okay, just like I said before, this ring puckering, we will talk more about that in class. Next, I want to address this word right here, complementary. What does that mean? And what that simply means is that we know G pairs with C and T pairs with A. That's just how it works. So if we only knew one of the strands, so we have this DNA molecule right here, and let's just say in that DNA molecule, for some weird reason, we only know one of the strands. Shown there. And we know the sequence, CAATCGTCA. Would we be able to figure out what the other strand is? Yes, so easy, because we know it's complementary. So all we would have to do is be like, okay, what's the sequence of the other strand? Well, if this one's C, then this one has to be G. If that's A, that has to be T, because we understand the base pairing. And so that's going to be very important when we start talking about biochemical techniques of trying to figure out an unknown DNA sequence. So if you're just given this random DNA and you want to know the sequence of it, you are going to use this principle of complementarity to figure it out. And so PCR and Sachinger sequencing are techniques that utilize this principle right here to help us figure out DNA sequences. Okay? And so we are going to talk about Sachinger sequencing and PCR, and you are going to learn how to use those techniques. But before we jump into some lab techniques, I want to continue our conversation of DNA and RNA a little bit more and just give you just expose you to like what other things nucleotides and nucleosides can do other than just DNA. So let's talk about RNA for a bit now. So that first bullet point, when you transcribe DNA, you're going to get a single-stranded RNA molecule. Okay? So DNA is double-stranded, RNA is single-stranded. Okay? We've already talked about the second bullet point. What's the difference between DNA and RNA? Okay. Well, you know, DNA is deoxyribose and then RNA is ribose. And then another key difference is that in RNA, we are replacing thymine with uracil. So now, when we look at what's going on here, so let's see, in DNA, it's A to T, that's the base pair, but RNA is going to be A to U. Okay. So let's jump in and take a look at a few cool features of RNA. And even though they're single-stranded, they kind of have a lot of cool structures that DNA doesn't have. Okay? So here's just a single-stranded RNA molecule. But what's going to happen here is this right here. Okay? There's some unique sequences that we can see in RNA here. So in this slide, what I want you guys to understand, this molecule up here, what it represents. I want you to think of it as RNA. Okay? And in order to really do that, we have to kind of fix the slide a little bit. So what you would do is just replace all the T's with U. Okay? So here we have a single-stranded RNA molecule. And what's cool about RNA is that it can fold in on itself in some unique motifs. Well, not motifs, just unique structures that we have given particular names to. So you'll hear about loops, and I'll show you what a loop is in a minute. But in this slide, we see a hairpin, oftentimes called a hairpin loop. And you can see that we're going to have these base pairs right here, so that would have to be a U and so forth, right, to make it RNA. Right here, look at that. That is a single-stranded RNA molecule, but it's just folding in on itself and creating these really cool structures. And it's these structures here that help give it its function. Much like proteins, if proteins are not in the correct three-dimensional shape, they lose their function. Another cool feature about RNA is when you look at these hairpin loops right here, if we can zoom in right there, you can see G to C, G to C, A to U, right? You can, when you get your individual slides, you can zoom in and look at that. You'll see that they're base pairing based off of what Watson and Crick told us. Okay, so Watson and Crick base pairing. But then in this molecule here, you could see, hey, G pairs with U. There's another G and U. And you can see in the graphic, it's not a line indicating a base pair, but a dot. And what's going on here is that G and U are still hydrogen bonding, just not in the Watson and Crick way. Okay, so these nitrogenous bases, they're going to turn and rotate in such a way as to hydrogen bond differently than what we are used to in DNA. And so you can see G should not pair with U, but it can. Okay? And so that's a cool feature of RNA, that it allows that to happen, and then that, I would assume, influences the secondary structure of RNA. Now, just another really cool example of the three-dimensional structure of RNA would be tRNA. Right. What does tRNA do? Well, it's just an RNA molecule, linear strand, that's all wrapped up into a three-dimensional structure that kind of looks like a letter T. And what do tRNA molecules do? Well, they bring the amino acid to the ribosome so to make a protein. That's its main function. Okay? So RNA, that field of study, is so interesting and so cool. All the different types of molecules that we can use, or biology uses, to keep cells alive and functioning properly. Next, what I want to talk about is looking at nucleotides and other functions that they can have. So we've been looking at nucleotides in DNA and RNA. Now let's kind of introduce what else they can do. So we have coenzyme A. And you can see right here in the peach looking color, hey, there's our nucleoside. It's a different looking one because we have our adenosine right there. We have at the 3' a phosphate group, and then all those, the phosphates right there. It has hydroxyl at the 2' position. So, and then you see you have this big fatty chain right here. And then we have a thiol at the end. So this molecule has a lot of components to it. Now, what is it used for? Okay, so you're going to see this a lot when you start looking at the citric acid cycle. Because in the citric acid cycle, what's going to happen here is we're going to, well, let's see here. For that one, what's going to happen is that this molecule is going to attach an acyl group. That is not correct. Let's fix that. We will attach a carbon like this, CH3, like that. Okay, so we have this acyl group. So get rid of that hydrogen. Now it's just the sulfur attached to the carbon right there. And now what you've seen this before. Okay? You have seen acetyl, or now we would call it acetyl coenzyme A. We'll just call it coenzyme A with this acyl group attached. Okay? And you've seen this when you have taken this species right here. Oxaloacetate, that is an intermediate in the citric acid cycle. And you're going to take that and convert that into citrate. Okay. And what's happening here is that the coenzyme A is going to transfer the acyl group. It's going to transfer that acyl group to the oxaloacetate to convert it into citrate. That's what's happening. So if you look at the structure here of oxaloacetate, we can look at it like this. Oh, I'm not going to have enough room. Okay, so there's oxaloacetate. And what does citrate now look like? Well, we're going to be tacking on that acyl group. And it's going to look like this, the way I'm drawing it here. Right there. Like that. So what has happened is this acyl group right there, what we've done is attached it right there. And you've seen that in biology class when you've studied the citric acid cycle. Okay. Now, when you look at NAD plus and FAD, you also seen those molecules in the citric acid cycle. And both of their purposes are that they're oxidizing agents. So they are going to help bring in electrons and move around electrons and hydrogens. Okay? And so if you want to look carefully at an example of FAD, you would look at succinate getting converted into fumarate. That uses FAD. If you want to look at an NAD+, an example would be alpha-ketoglutarate getting converted into succinyl-CoA. So I bring this up to you because I want to help you guys make connections that you have seen these molecules in action, but you probably up to this point has just seen them in blobs of circles or squares or stars. But these molecules that you've studied in the past have structure to it. And you can see that they're all coming with these nucleosides or nucleotides. Okay? What's highlighted is just the nucleoside. Okay? Because remember, nucleotides come in with the phosphate. Okay? Do we have some other ones here? Okay, cyclic AMP, that's going to be used in signals. Like when your cells encounter something, let's say glucose. And so glucose needs to be processed. So insulin is triggered and all this good stuff. The cells need to know what's going on, and cyclic AMP is one of those signaling molecules to help the cells get ready for what's coming. Okay? And so all of these you can see are just different variants of nucleotides. So it's pretty cool stuff, and you're going to see these molecules in action in upcoming chapters. So I think that's a good place to stop. And as always, if you have any questions or concerns, please reach out. I'll be happy to help.